In 1952, bacteriologists Alfred Hershey and Martha Chase attempted to find out whether it was proteins or DNA that contributed to the genetic makeup of cells. To conduct their tests, the bacteria they used was E. coli, and they infected it with a virus called a bacteriophage. A bacteriophage is a virus that infects bacteria specifically. At that time, scientists knew that bacteria could be transformed by viruses, but they didn't know which part did the transforming, the protein coat or the DNA. When phages infect bacteria, they attach themselves to the surface of the bacterium via long tail fibers and inject their DNA into the bacterium cell. Once the host is infected, reproduction is able to occur. To establish whether the phage injected protein or DNA into the cell, Hershey and Chase prepared the phages in two different medium. The first batch was produced in a medium containing S35, a radioisotope of sulfur. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA, since sulfur is found in proteins but not DNA. The sulfur-labeled phages were then allowed to infect the bacteria. They attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. As a result, the phage produced in these cells contained no radioactivity. The second batch of phages were produced in a medium containing P32, a radioisotope of phosphorus. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein since phosphorus is found in DNA but not in phage proteins. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in subsequent phages that were produced by the infected bacteria. For the sake of simplicity, only a few subsequent phages are shown. In reality, they multiply rapidly within the bacterium until the bacteria lysogenates or bursts, causing the phages to look for a new host. After both batches of phages infected the bacteria, each culture was mixed in a blender to remove any phage parts remaining outside of the cell. Finally, the cultures were centrifuged, which causes heavier materials, such as bacteria, to move to the bottom of a test tube and form a lump called a pellet. Lighter materials, such as the medium used to produce the phages, remains near the top of the tube, forming a liquid layer called the supernatant. By measuring the radioactivity in the pellet and supernatant, a large amount of P32 was found in the pellet or the bottom, and almost all the S35 was found in the supernatant or top liquid layer. Since the P32 was found in the pellet, they concluded that DNA and not proteins functioned as a genetic material of the phage.